I'm Ivano Legnini. I'm a group leader in the Genomics Research Center at Human Technopole.
Hi, I'm Linda Masante. I'm a postdoc in the Legnini Group.
Hi, I'm Anna Kotte. I'm a PhD student in the Legnini Group at the Genomics Research Center of Human Technopole.
My group is interested in many different things, otherwise we will get bored. I would say that all our research questions orbit around the problem of gene regulation. We're interested in the mechanics of it, how it works, what's the exact series of events that an RNA molecule experiences throughout its lifetime, and why do different RNA molecules have such different lifespans. And if we understand this, can we use this information, for example, in designing better mRNA vaccines or drugs? But also we're interested in the effects of gene regulation, for example, in development or in neuronal function.
I study the RNA metabolism at different layers, so from transcription to degradation, specifically in neurons. And I study in particular a group of proteins named CPBs proteins and their possible roles in the orchestration of neuronal RNA metabolism.
My research focus is in understanding regulatory principles that drive mRNA metabolism. Now, we know that during their lifetime, mRNAs are subjected to some general metabolic pathways, but what's interesting is that these same metabolic pathways can act with very distinctive dynamics and outcomes on each mRNA. And we still don't fully understand how these molecular machineries could possibly distinguish between all the different mRNAs. So my goal is to try to find globally the information and the features that are required to distinguish between these mRNAs and determine their fate once they enter these metabolic pathways.
I think HT is unique in the diversity of backgrounds and expertises that you can find here, and especially the density of it. We're not a huge institute actually, but you can find all kinds of research questions from structural biology to molecular biology, genomics, large population genetic studies, and so on. And I think all of this contributes to fostering an extremely interdisciplinary approach, which is also what we try to adopt in my own group. For example, it's reflected in its composition. We have molecular and cell biologist, biochemist, an engineer, a student in bioinformatics, and so on. And I think all of this contributes to make better science.
The best opportunity that HT has given me so far is the freedom to choose the research project that I'm doing for my PhD. And freedom in research is not something to be taken for granted, especially for a PhD student.
In HT, I would like to exploit the highly different backgrounds of the different centers. So I would like to start and carry on a project that is multidisciplinary and do it exploiting all the technological availability that we have in HT.
So the most exciting aspect for me in working at HT is being part of a very collaborative and interdisciplinary team of scientists which constantly exposes you to new mindsets and approaches to research.
The moment you get here, you realize you have access to an incredible playground of new technologies, but also an incredibly young and fresh environment. And plus, people are very kind.
The most exciting aspect of HT so far is the constant feeling of collaboration and connection among members of different research centers.
So HT for me is innovation, exploration, and collaboration.
HT for me means expertise, collaboration, and complementarity.
Excellent science, exciting collaborations, and ping pong.